Well, analytical sciences and engineering is a bit of a departure from the way the field normally thinks about chemical measurement science. Here at Notre Dame, we had the opportunity to build a program from scratch. So we took that opportunity and developed a plan which incorporated all the important intellectual drivers behind the fields like biology, chemical engineering, electrical engineering that go far beyond the confines of a chemistry department. We have a model of organic growth. So it's been a really powerful way to assemble a close-knit group of scholars. Norm Dovichi coming to Notre Dame was really a seminal moment in our history. Norm is a world-leading scientist. His role in developing the technology that ultimately sequenced the human genome, I think, is the thing most people know him for. He's now moved well beyond the genomic problems that he began with to work on new avenues for understanding protein expression and the products of protein expression, which are the metabolites from cellular chemical reactions.
We started working in the Human Genome Project in the late 1980s, and our goal was to develop high-throughput capillary array electrophoresis instrumentation. Upon completion of the human genome, and my group was interested in proteomics, developing advanced tools to characterize protein contents of tissues. An interesting example comes from a recent collaboration with Paul Huber, a biochemist in the department. And Paul is an expert in a wide range of biological sciences, but in particular in Xenopus laevis biology, that's the African frog. Our goal is to characterize the protein expression changes that accompany development of a vertebrate brain. Now we're up to over 5,000 proteins at 6 different stages of development. We have identified those cells whose lineage includes the Xenopus brain, and I've tagged those cells with fluorescent markers, isolated those cells from the individual embryos, and are preparing to do proteomics on those samples. And this is cool because we'll be able to understand how nature goes from a pluripotent cell, a cell that can do anything, how it chooses its fate of its cells during development, steers those cells into specific developmental pathways, and to understand how the brain is assembled in that organism. Because by doing so, we can understand, of course, normal development, but more importantly, abnormal development, which has profound implications in healthcare. Usual chemists work in the field, and I work in their silos. Engineers work in their silos, and the two communities tend not to communicate very effectively with each other. And Paul Bohn had the vision of getting the two groups to work together, talk together, meet together, and collaborate with each other.
Some of the areas that I study are plasma science and engineering, so how to generate and manipulate plasmas. When I came to the University of Notre Dame, I was very fortunate to open up collaborations with faculty such as Chia Chang and Norm Dovichi and Mario Lieberman, where we wanted to explore developing new tools or new ways to conduct mass spectrometry, particularly ionization techniques. I was able to grow into this area of chemical analysis, and now I continue to integrate it into many areas of my research. And it has also created opportunities for me to engage with chemists who have a more chemical perspective, and I can bring more of an engineering and especially a transport perspective. How do these things move? How do we manipulate these things on very small scales before we detect them? Which is very critical for things such as concentrating them or identifying very minute quantities. One of the reasons that really attracted me to this institution was that it gave me a lot of opportunity to conduct research without bounds, to really pursue questions that I was interested in. And along the way, I find new questions that I'm interested in, and I can pivot or change directions. And so when I started the university in 2008, I had a vision for the type of research I had, but it's actually not exactly what I'm doing today.
Coming to Notre Dame gives you the opportunity to work in an extremely exciting environment. Not only are engineers working with chemical scientists, with biologists, but we're doing this in a way that allows a growth path. So this ability to work across disciplinary lines, to do it in an integrative and natural way, is one of the very exciting things about Notre Dame.